Hi to anyone who's just joined. My name's Evie. Welcome to Researcher's Stories. I'm the research assistant for the Butterflies Through Time project. If this is your first time, this is our fifth talk in the series, where each week we listen to a new butterfly researcher, talk a little bit about their research and what it means for the wider butterfly conservation. So the series has been running every Wednesday at 7 p.m. and it's going to be running until the 8th of June. So this is the second to last in the series, and our last one, it will be next week on the 8th of June. So cool. So today we're going to be hearing from Esme. So Esme is a PhD student in the insect ecology group at the University of Cambridge, and she works alongside the Wildlife Trust of Bedfordshire, Cambridgeshire, and Northamptonshire. So her research is focused around the effects of temperature on butterflies and to better understand how they respond to climate change. So in a moment, she's going to talk a bit about her work understanding temperature's role in the small blue eggs distribution and how this can inform management on nature reserves. I think that's it from me, Esme. You're free to take over. Over to you.
Hi, everyone. So I'll just start sharing my screen. You may notice I sound a little bit croaky. I'm afraid I lost my voice a few days ago, so hopefully you can all understand me, and I really hope this very smart captioning software can keep up with how weird my voice sounds at the moment. But anyway, thank you so much, Evie, for that little introduction. Hopefully you can all see my screen. My name is Esme Ashe Jepson, and I'm going to be talking to you today about my research that is all focused on the opportunities and challenges of researching caterpillars, in particular of butterflies. And all of my research falls under the title of Cool as a Caterpillar, investigating the effects of temperature on butterflies. I'm going to be talking you through some of the progress I've made, some of the challenges I've faced, and we're going to end with a nice little case study of how my data can be used on the small blue butterfly. That's what you can see on your screen right there. So firstly, a little bit on who I am. So I'm a third-year PhD student in the insect ecology group at Cambridge. All of my research is done in collaboration with the Wildlife Trusts of Bedfordshire, Cambridgeshire, and Northamptonshire. And normally, when I'm not out in the field crawling around looking for caterpillars, I am based in the museum. So you may have seen me around there if you've ever visited. You may also have seen me doing a little bit of outreach in the museum as well. And we actually have some plans over the next couple of years to do some more outreach. So you may hear more from me in the future. My background is in conservation and ecology. My research in the past has focused on climate change, insect evolution, and occasionally population genetics. I want to dip my toe into something a little bit different. But now all of my research is focused on butterflies. So I thought I'd start with why should we even care about butterflies in the first place? Well, firstly, conservation and nature reserve management may change in the face of climate change. As we know, temperatures are changing globally, in particular in the temperate regions of the world where we are here in the UK. And at present, we have lots of beautiful, diverse communities of butterflies in our nature reserves that need to be managed so that they can continue to be effective. But as the temperatures are rising, you may know that some species are changing their ranges. For example, here in the UK, a lot of species have started moving northwards. And unfortunately, that means eventually they may end up leaving our nature reserves, because unfortunately, our nature reserves can't move north with them. And that means we need to think of smart ways to manage our nature reserves so that they can continue to be as effective as possible, no matter how much the climate may change. So butterflies in particular are a really interesting and important group. They are very sensitive to temperature. There's a huge background of information available on butterflies, and that means that we can ask really nuanced questions and can continue to collect data that can help us answer questions that span really long time spans. They have very short generation times, which means essentially they breed very quickly, and that means they're very sensitive to any changes. You can pick up these changes in the butterflies very, very quickly, which can be useful if you're thinking about other species as well, who helps to protect. And protecting the environments that butterflies live in also helps to protect all the other organisms that live in those environments. Butterflies themselves are also an important food source for other species, particularly as caterpillars. They're also pollinators as well. But sadly, many butterflies are declining. About a week ago, a new report was released showing that 50% of British butterflies are now on an extinction red list. So our butterflies are declining and they need help. But what do we know about butterflies? Well, we already know quite a lot, like I said, but that information generally falls on the adult life stage. That's only one out of the 4 life stages of egg, caterpillar, pupa, and then adult. What about all those other life stages and their responses to temperature? And you yourself may have noticed that, if you've ever visited the museum, that the vast majority of butterfly specimens are of the adults rather than the other life stages. So thinking more about what we already know, this is a little plot that's coming out of some of my research at the moment, where I'm investigating the published data all on butterflies and the effects of temperature on them. Each bar represents a different life stage. So on the left, you have egg, then caterpillar, then pupa, then adult. You don't need to worry too much about the different colors. Those just refer to the different butterfly families, of which there are 6. And what we can see from here is that the vast amount of data coming out on butterflies and the effects of temperature on them are on the adult life stage. In fact, if you add all of the other life stages together, you get pretty close to how much data is coming out on the adult. So we know quite a lot about the responses to temperature, but limited really to just one life stage. And also one particular family, Nymphalidae, this blue one here. So my goal is to address this gap in our understanding, primarily of caterpillar responses to temperature, as they're obviously able to move around and they have different behaviors that can make them slightly more dynamic in response to temperature than, let's say, the other life stages. Although my research also does focus on the egg in some cases. I generally don't work on the pupa. We'll get into why in a moment. So does it matter if we only know about one life stage? What if that's informative enough? What if that tells us everything? Well, unfortunately, that is not the case. Studies have shown that temperature may positively impact one life stage and negatively impact another. So though temperature may positively impact the adult life stage, that may make us think, oh, this butterfly is going to be fine in climate change. We don't need to worry about them. All of our resources can go to other species. Whereas actually, if you looked at the caterpillar, it has a much stronger, much more negative impact. That means that we really should be concerned about that species. So only studying one life stage solves only one small piece of the puzzle of what that species is going to do under a changing climate. So why caterpillars? Because that is what most of my research is conducted on. Well, this is the life stage that gathers pretty much all of the energy needed for most of the life cycle. So they're entirely dedicated to eating, which let's be real, that's not a very bad life. And what they do is they convert plant material into energy for growth, development, metamorphosis, and in a lot of cases as well, reproduction. So any changes to the caterpillar life stage is going to affect all of those other aspects of their biology. They're also an incredibly important food source for many other animals, specifically as caterpillars. So you may have heard many nesting birds will time when they lay their eggs and when they're feeding their young to when the caterpillars are around, not necessarily just of butterflies, but also of moths. They're an extremely important food source for birds such as the blue tit, for example. Many caterpillars are also very important global pests. So it could be very interesting to hear what might change under climate change with these species. But as a life stage that are dynamic, that are able to behave in certain ways like the adults, they're extremely vulnerable. They're not very mobile. They're often trapped essentially on a single specific food plant that they can't leave. So if they are at the mercy of the environment, at the mercy of the temperature around them, it's very important for us to understand how they're going to respond to these changing temperatures. They're much less able to take advantage, for example, of different microclimates. They can't fly off to find somewhere nice and cool when it gets too hot, and they can't escape when it gets too cold to go and find somewhere to bask. They don't have those beautiful giant wings that they can use like solar panels to warm up. They're just these small little caterpillars. So I thought I'd mention maybe a few ideas on why there aren't more studies on these other life stages. Well, adults, as I said, really dominate the literature on what we understand of the effects of temperature on butterflies. This could be because they're very pretty. So it's very easy to get people interested and engaged, which is hugely important. They're also very easy to find and identify from a distance. You can just walk a little transect through a beautiful field and you can count the butterflies from a distance. be able to identify them maybe to a species level. Often you can even sex them from a distance as well. Super easy to collect this data. But what about the other life stages? Well, eggs are very small and often very hard to find. And if you're not sure what species laid that egg, identifying it just from the egg to a species is very difficult. For caterpillars, they can also be very hard to find, and they can often be hard to identify. Many species look very similar to each other. In my personal experience as well, they're also very good at running away from you. Once they've realized that you've spotted them, they are surprisingly fast, and they are wriggly, and they get away from you, drop into the grass, and they're gone. Pupa, on the other hand, are incredibly delicate and often very well hidden and camouflaged to their environment. Me personally, I've found pupa, the life stage, the least often when I am crawling through these meadows looking for butterflies. They're incredibly well hidden, but they're also very, very delicate, so I prefer not to mess with them when I get the chance anyway. For these other life stages, we can study them in a lab, but that raises the question of how relevant are the results we're going to get out of a lab to these complex interactions going on in the real world. And that's what I'm really interested in, is the ecology and interactions going on. So here we have a kidney vetch flower, which is what the small blue lays on. And there is an egg on this flower, just to demonstrate how small these eggs can be. I'll give you a few seconds to see if you can spot the egg. Just a few seconds, and there it is. Tiny, tiny little speck there. That's where the eggs, that's what the eggs of the small blue look like on these flowers. You can see this ruler for scale, how tiny these eggs are. And also for the caterpillars themselves, this is a lovely little shelter built by a dingy skipper caterpillar. And what they do is they roll the leaves of bird's foot trefoil, which is a small plant. Each of these leaves are barely a centimetre, and they live and feed hidden inside these shelters. So they're incredibly hard to find. So all of my research comes from 4 study sites in Bedfordshire. So here we have Tottenhoe Quarry and Tottenhoe Knolls. We have Blows Downs and Pegston Hills, and these are beautiful sites. You can see here what they look like. So these are all calcareous grasslands, which means they're mainly chalk. You can see how white the bare ground is. They're very chalky, which means they're very low nutrients, which allows really rare and specialist plants to thrive. You can see how diverse the floral communities are on these sites. They are beautiful. If you get a chance to visit them, I would recommend it. And because there's so many rare and diverse plants, there's a lot of rare and diverse insects as well that are on these sites. So they're a great place for me to go and study lots of different species, all cohabiting in one area. And you can also see here in this shot how lumpy bumpy these reserves are. They're very topographically diverse, which means they've got lots of north-facing slopes, which are nice and cool, that I can compare with south-facing slopes, which are much warmer. So that gives me a way of investigating how these caterpillars can use these different thermal environments. So I'd mentioned a little bit about these sites and the importance of doing conservation on them. So grasslands are declining and unfortunately need protecting. Only 1% of the land area in the UK is grasslands, and we have lost 97% of our wildflower meadows since the 1930s, often for agriculture. That is equal to 1.5 times the size of Wales. But grasslands are not only important for lots of these rare species that I've mentioned, they're also very culturally important to us. They're a part of our heritage here in the UK. There are over 2,000 roads in Britain containing the words meadow or dol, which is the Welsh for meadow. And they're home, as I mentioned, to lots of rare species of butterflies. So for example, the Duke of Burgundy and Chalk Hill Blues. And I've included here a little quote from Dr. Trevor Dines, who's a plant life botanical specialist. The steady, quiet and underreported decline of our meadows is one of the biggest tragedies in the history of UK nature conservation. If over 97% of our woodland had been destroyed, there'd be a national outcry. Without the roar of chainsaws or the sound of mighty oaks crashing to the ground, meadows with undisturbed floral histories going back generations are being ploughed up in a single afternoon. So I personally feel that not only are these species interesting, but the habitat that they live in is precious and it needs to be managed correctly. So here are some images of all the different butterfly species that I am working with. As you can see, it's very diverse, lots of colors and sizes and shapes, but who cares? These are the adults. No one wants to see that. Here we have them as caterpillars. As you can see, they are all very diverse in colour, in form, in ecologies and behaviours as well. There's a lot of diversity going on here, and all of these live on those sites. So my research can broadly be split into 5 main topics. The ones in bold are the only ones I'm going to be discussing in detail today, but I thought I'd mention the sorts of research going on in the museum and in the insect ecology group with butterflies. So that first one is leading on, if you remember that bar chart from earlier, it is describing what we already know about the effects of temperature on butterflies. The purpose of this is to identify biases and gaps in the literature that we can address. Secondly, it's about microclimate temperatures and environmental characteristics that may be influencing caterpillar distribution in nature reserves. By which I mean, where are these caterpillars in the nature reserves? Why are they over here and not over there? And on a much finer scale, why are they on this one plant and not this other plant right near it? What is it about the plant that they like? And that will lead into the final point, but we'll get there when we get there. The next part is on buffering air temperature. So this is asking, are caterpillars able to buffer themselves against changes in air temperature? And how does their ability compare to adults? So that's about comparing these different life stages. Another part of my research is comparing temperate butterflies and tropical butterflies and their buffering abilities, but I won't be talking in detail about this. And lastly, a case study with the small blue on their egg-laying behaviour and changes to their emergence through time. So digging a little bit more into what exactly I do to answer these questions, firstly on the microclimate temperatures and environmental characteristics that influence their distribution. First, we need to understand the food plant preference. If they're only laying on one plant, then we need to understand what's going on with that plant first. So for example, let's say we have the peacock, which lays on stinging nettles, and we go out and we just look at where the caterpillars are. And it looks like they're all on nice, sunny, south-facing slopes. We can say, oh, that's great. We need to protect south-facing nettles, and that'll help the breeding of the peacock butterfly. But what if it turns out that nettles just happen to only really grow on south-facing slopes, and actually the caterpillars were primarily on slightly more southeast-facing slopes, and those are the ones we need to protect for the butterfly. So once we have the food plant preference, we can then understand how the caterpillar preference aligns with this. So here we have 3 plants. I'll take you through them. Here on the left is elder. This is the food plant of the brimstone. Here in the middle is garlic mustard, the food plant of orange tips and also green vein whites. And on the right, we have those stinging nettles. So this is the food plant of peacocks, small tortoiseshells, red admirals, and also commas as well. So what I end up doing is marching around these sites. Here is a map of Tottenham Knolls, and I have mapped the location of all of these different plants on that site. So yellow is stinging nettles, red is elder, and this one little blue point is the only garlic mustard on the site. So that's not particularly useful in this site, but interesting that there is just the one. And what I do for every single one of these points, I've described the area around it. What sort of a slope is it on? How big are these patches? And then I go back, I revisit all of these points and look for caterpillars. And the way that I look for caterpillars changes depending on how hard they are to find. So I have 2 methods. I have suction sampling and focused searches. The trick is finding them. So let's say you need to find this little guy. This is a meadow brown caterpillar, a very common butterfly, and it feeds on grass. You need to find that in a grassland. That's going to be a little bit tricky unless you fancy crawling around these meadows hours and hours on end looking for these caterpillars. That's just a waste of time. You're not going to be able to find enough to really understand what's going on. So what I use is a neat little piece of technology, this here, which I call my suction sampler or GVAC. All it essentially is an inverted leaf blower, where instead of blowing, it sort of hoovers the grass. Don't worry, there is a net in this end to catch all of the insects and grass and debris that I hoover up within certain locations within these nature reserves. I then empty all that out into a tray and I dig through looking for caterpillars like this. Here's one that I found. This is a little ringlet caterpillar, another one that just feeds on grass. So that's a very useful method for looking for grass feeding caterpillars in particular. The next type of surveys that I do are focus searches, which is really useful for species that feed only on a specific food plant. So I just need to look at that food plant. Again, here we have kidney vetch, and actually there are 2 caterpillars in this picture. I'll give you a little moment to see if you can spot them. Just a few moments. It's the small blue we're looking for here. And there they are. So you have one sort of greenish one here and one sort of pinkish one here. And what I do is I search these all by hand. Here you can see my little biodegradable markers. This is just some undyed yarn that I end up removing anyway at the end of the survey. And I mark each plant that I've searched. You can see them all here. See, this one has a black mark. That means there was a caterpillar on it. This one doesn't have a mark, so there was nothing on it. And I record lots of information about the plant itself and the environment that it's in. Another piece of data that I collect is temperature over time. That's what this little thing here is. This is a data logger, which records temperature every hour over a couple of months. And that helps me understand the thermal environment that these plants are in and the caterpillar itself is in. Right, so that answers that, sort of gives you a little bit of an insight into how I look into that question about how these caterpillars are distributed in these nature reserves. But there's also more data that I can collect once I've found them. And that helps me answer this question of how these caterpillars are able to buffer themselves against changes in air temperature. I'll go in a moment into what I mean by buffering. So this is during the focus searches. I use this little piece of tech here, which is connected usually to a big handheld indicator. And this is essentially a very sensitive probe, and you gently press the tip of this to the side of the caterpillar, and it tells you in very, very precisely what their body temperature is. And then I can record also the temperature of the plant that they're on and the air temperature that day. And that helps us understand how their body temperature changes on a cold day versus a warm day. So when I'm talking about buffering ability, what I'm talking about is the relationship between their body temperature and the air temperature that day. So I've included 2 graphs here. On the left is an example of a bad buffering ability, this blue line, a very steep line, versus on the right, a good buffering ability, a shallow slope. And what I essentially mean is in the relationship between body temperature and air temperature, this black line here just shows a one-to-one relationship if their body temperature is always exactly the same as the air temperature. And usually we find this is not the case. They are doing something. That means that their body temperature is either warmer or cooler than the air. But if something is very bad at buffering, it essentially means that they are experiencing a very wide range in body temperature over quite a narrow air temperature range. It means when it's cold, they get very cold. When it's hot, they get very hot. And caterpillars are sensitive to temperature. If they get too cold, they can't feed, they can't grow. If they get too hot, they might die. So this is not good. This shows it may be a bit of a problem if temperatures continue to rise. Their body temperatures are not controllable at high air temperatures. There's a good chance that species is at risk. Whereas on the other hand, a good buffering ability shows a really narrow slope, where essentially at a low air temperatures, their body temperatures are nice and warm. They're warming up on a cold day. Whereas on the other hand, on a nice hot day, they're cooling down. So they're much less at risk of this sort of temperature damage that they might experience. They're able to maintain quite a narrow range in body temperature over a really wide range of air temperatures. And perhaps we don't need to worry too much about those species under climate change. So now to share just a few very early results. I haven't finished looking at this yet, and I'm continuing to collect data this summer. What it looks like here in this plot, this is again the same, it's just body temperature against air temperature. The red dots and the red line are adult butterflies, and blue are their caterpillars, and these are of the same species. And what you can see is that the red line is much shallower. There are much better buffers than the much steeper blue caterpillar line. And what we have found is there is a significant difference between these life stages. Adult butterflies have a much better ability to buffer air temperature than caterpillars. And it appears that certain traits seem to be particularly influential for caterpillars in particular. So for example, if you remember that little dingy skipper shelter I showed you earlier, the species that are building little shelters like this are much better at buffering air temperature than caterpillars that don't. And it may be because these shelters are allowing them to build this nice cool area to stay in when it gets a little bit too hot, and they're able to crawl out and warm up when it gets too cold. Colour also seems to be important for caterpillars, where pale species are better at buffering air temperature than dark species. Interestingly, this is exactly the same pattern found for adult butterflies, where pale adult butterflies are better at buffering air temperature than dark adult butterflies. And lastly, gregarious species seem to be struggling more. And what I mean by that is I have a little video for you here. We'll just play this really quick. Don't need any sound. So this writhing black mass is peacock caterpillars who live all together sort of as a defense mechanism. They all stay together right at the top of a stinging nettle plant, and they build a web. You can just about see the web here. And they all stay together. And if they're disturbed, they start flicking around, and they're very frightening to any predator that comes along. They're able to defend themselves very well because of that. But you may also notice that they're also quite dark, and they're right out in the open at the top of the plants, and they're one giant dark mass. And what it essentially means with this gregarious pattern is that species that clump together like this as caterpillars, when it starts getting a bit hot, they're very reluctant to move away from the group, to go and seek somewhere cool to cool down. They just seem to sit there and suffer. Let's move on to, if I can get past this slide. There we go. All right, so I've actually got the plot here specifically for peacocks. Here on the left, I thought I'd show you two little examples of specific species and the different things that they seem to be doing. So here you can see how steep the slope is for peacock caterpillars, again in blue, versus the adult in red, which is a very shallow slope. They're very good at buffering air temperatures, potentially because they're lovely, they're big and strong, and they're able to fly out and seek these warm and cool places. Whereas these caterpillars are stuck together, exposed at the top of these stinging nettles, all clumped together and also very dark in colour. Everything seems to be conspiring against these caterpillars. So even though if we'd just taken a look at the adult life stage, we would think, oh, this species is fine. We don't need to worry about them under rising temperatures. But actually, it looks like they may struggle to complete their life cycle under increasing temperatures. We can compare this to the small white here on the right, which again, the caterpillars are in blue and the adults are in red. And what we can see is that there is no significant difference between the two slopes. They're very similar, and they're actually both pretty shallow. They're both pretty good at buffering air temperature. They're both quite pale as far as it goes. As far as caterpillars go, this is pretty pale. They also live by themselves. They're not gregarious, and they're able to seek shelter when they need it. Something that is interesting about the small white is it is an invasive global pest of agriculture all around the world. Maybe this is why it has been such a successful pest, because it is able to buffer its air temperature very well throughout its life cycle. It is able to complete its life cycle without much struggle, no matter what the climate is, whatever country it ends up in. It's food for thought. So now, what can we do with all of this sorts of data? Well, the purpose of my data, of my research, is to help inform nature reserve management and the conservation of butterflies. And I thought I'd talk you through a case study of the small blue butterfly. So this here is the small blue, a lovely little thing. It is our smallest resident butterfly, teeny tiny. It's also a food plant specialist, which means it only feeds on one plant, the kidney vetch, which makes life a little bit easier for me. It means I just need to search kidney vetch plants. Something that I find interesting about this species is that the larvae of the caterpillars are cannibalistic. So it does not matter how many eggs are laid on one flower, only one caterpillar will survive. And that tends to be the first one laid, the oldest or the biggest. Not always, but it tends to be. And they've also unfortunately undergone a decline over the last 40 years. They have a very patchy distribution, but they do seem to be quite stable, but very patchy. And though these very small populations can be very easily lost. So I wanted to answer the question to inform these nature reserve managers where they're laying their eggs so that we can protect those areas. So what we did is we collected data over 4 years, covering a 14-year period to work out if any of this is also changing over time. This involved focus searches of kidney vetch for the eggs in particular, And I was recording characteristics of the plants and their surrounding environments with the goal of identifying a preference so that these nature reserve managers can help manage their nature reserves to prevent the loss of this declining species from their sites. So the fieldwork looked like this. I've already shown you one of these. There is again an egg on this flower. This one should be a little bit easier to find. It is there, this tiny little thing. It's like a little donut when you get up close. And I have a few results I'd like to share with you. So the first little piece of results is about when they're emerging over time. This is when they're very first coming out of their cocoons and the adults are flying about. And this helps us understand how their process of development is changing over time. And what we found were two things. Firstly, here on the left, graph A, was that their emergence is getting earlier over time. We have year here along the bottom axis and day of the year on the y-axis. And essentially what we find is that they're emerging roughly 2 days earlier per year. And what we also found was that their emergence is earlier in years with a warm February. It seems to be February is the month that is important in the development of this species. It could be that this is triggering them to wake up from hibernating over winter and they start developing quicker. We're not quite sure exactly why, but this is the month that seems to be important for them. Next, we wanted to look at whether their abundance has changed through time, how many people are seeing each year. And what we found was that their abundance could not be predicted by temperature. Now, something that is interesting about small blues is that they have extremely variable abundances between years. So we have this quite nightmarish looking plot here that I'll talk you through. So the black line, which is the collated index of abundance, what this essentially means is the number of small blues seen at the national scale in the UK, at the whole scale of this whole country. And you can see it has these characteristics, peaks and valleys, peaks and valleys, peaks and valleys. And this is pretty consistent over time, just peaks and valleys all over the place. And this gray line is the data from our study sites. And you can see it matches quite well. You have these valleys and peaks, and valleys and peaks that match with the national trend. So whatever's going on with this species to make it so variable year to year, it's affecting it not only in our site, but at the scale of the country. One thing that is interesting is our data was collected in 2006, 2007, 2008, and then I went out in 2020. Here we have in green 2006, red is 2007, and blue is 2008. What we have in our data is we've captured a peak and a valley year so that we can see if anything is different between these years, to see if we can pick out what is going on here. So now we're looking at small blue egg presence. This is where are they laying their eggs? And what we found was that small blues were laying their eggs on flowers that were taller and more apparent above the surrounding vegetation. I'll go into more detail about apparency in a moment. And we also found, that's this top row here, and we also found that small blues were laying eggs on flowers that were surrounded by tall vegetation. Both of these are consistent across all of these years. Unfortunately, we could not include 2006 in this particular analysis, though it comes in later, just because of the way the data was collected back then. So what I mean by apparency is this beautiful diagram that I have put together. Basically, that apparency is, this yellow thing is the flower head sticking up around the surrounding vegetation. This is how apparent it is, how much it's sticking out. And the caterpillars live inside these flower heads. They're also up above the surrounding vegetation. So the butterflies really like to lay their eggs on these sort of plants that are really high up above the surrounding vegetation and by nature also taller, versus these where the sort of flower is shorter than the surrounding vegetation, sort of sunken in a little bit. They don't like this negative apparency. They really like more apparent flower heads. So next, on to the next thing that we found that would influence where they were laying their eggs is solar radiation, which essentially just means how much sunshine a flower is receiving. So this might influence how the flower grows potentially, or how warm that flower might get. And what we found is it has a very sort of weak negative effect in 2007 and 2008, and a slightly positive effect in 2020. So this effect is differing weekly between years. So I'm hesitant to really talk too much about this. And the last thing that we found that seemed to be important for these butterflies is that they really like to lay eggs on flowers that had fewer nearby flowers to choose from. Essentially, there's less choice. So if there's less choice, they'll all lay or lay on the flowers that they have available. And this is also consistent across this quite wide time gap. Next, on to egg abundance. And the reason we've looked at presence and abundance separately is for the conservation of this species, we want to maximize egg presence. We want them to be laying eggs on as many flowers as possible, but we don't want them to be laying all of those eggs on the same flowers, because if you remember what I said, this is a cannibalistic species and they all end up eating each other until only one remains. It's a waste of eggs. And unfortunately, we found the exact same traits influence egg abundance as egg presence, and here we can include 2006. It's a little small and it's a little faint, but you may even see some of these points show over 15 eggs on a single flower. It must have been an outright war zone on these flowers compared to other years where it was relatively rare to have more than a couple of eggs per flower. So here what we found was that the small blues were laying more eggs on flowers that were taller and more apparent above the surrounding vegetation. This is consistent across all of these 4 years, including a peak year. And the same again, small blues are laying more eggs on flowers that are surrounded by tall vegetation. This is also consistent again across all 4 years, including this peak year. Excuse me. So what does this mean for management? So the small blues, firstly, we found are emerging earlier and earlier over time, but their abundance does not seem to have consistently changed over time, nor does it seem to be overly related to temperature, at least in the ways that we've looked at it. Because it is so variable, excuse me, one moment, just need to, just need to cough. Sorry about that. So what might be happening here, excuse me, I've lost my voice a bit. Excuse me, sorry about that. Hopefully you can all still understand me. So the abundance trends may be being hidden by how variable the abundance is year to year, if you remember that plot. So what we might just need to do is wait until we have more and more and more and more data that we can then look at more trends over time, over a greater time scale. It may be that there are trends going on there, but they are hidden by that variability. Next, for the preference for egg-laying sites, gosh, I hope you can all still understand me. Sorry about my voice. Excuse me. That these preferences are very consistent over time. That's really useful for management. It means that management can also be consistent over time. So with these egg-laying consistencies, the worry about this is that they are consistent between peak years and valley years, like I mentioned, which means in a valley year, that's not too bad. They are still laying quite low numbers of eggs across lots of different plants. But in peak years, these egg-laying females are not willing to compromise on where they're going to lay their eggs. So they all lay their eggs on a very small number of plants. And that may contribute to why we see such strong valley years after these peak years. It could be all-out war is going on in these flowers, and a very small number are actually surviving. What this tells us is just we need as many suitable plants as possible, and that should help them from stop laying eggs all on the same flowers. So management needs to aim to produce lots of very, very tall kidney vetch with tall surrounding vegetation. That should help reduce competition for this species. This can be done by protecting kidney vetch patches while they're growing and while these caterpillars are feeding away from cutting and grazing. Because unfortunately, both sheep and rabbits, as I found out as I was searching around for these flowers, really love to eat kidney vetch, which is a little bit unfortunate. So perhaps even just something as simple as a little fence up, or just delay the cutting until small blues have already started pupating or overwintering, may really help this species in those sites. So what's next? Well, as I said, I'm still collecting my data. So there is more data to be collected on lots of other species to identify where these butterflies are breeding in these nature reserves, to identify what these preferences are, so that we can then protect those areas in the future. I'm also going to be identifying specific species that may be at risk under rising temperatures, particularly with this buffering data, as I mentioned, and I've showed you a few little plots of that. And to complete our understanding of butterfly responses to temperature by studying the caterpillars, not just the adults. The final outcomes of my research, the goals, is to provide information that is usable for the Wildlife Trust and other nature reserve managers to inform the management of their nature reserves. I also hope to be doing some outreach in the museum over the next couple of years, which might raise some awareness and appreciation for these wonderful creatures, not just the adults, but also the really interesting little caterpillars as well. Overall, my final outcome, hopefully, will be improved and informed butterfly conservation. Thank you so much for listening. Thank you for making the time to come along today. I want to say a really quick little thank you to the Wildlife Trust of Bedfordshire, Cambridgeshire, Northamptonshire in particular, but also for Cambridge Conservation Initiative, CCI, and the Evolution Education Trust, EET, for their support as well. So thank you very much. A little bit of contact information there, should you want to reach out to me.
Brilliant. Thank you so much, Esme, for your insights into your research and the cannibalistic but fascinating small blue there. I'm not sure how many people knew that they could be cannibalistic, which is quite interesting, isn't it? If you love that. And I especially love how tiny the eggs are. It was incredible. How on earth did you spot that?
With much difficulty at first, but you get your way in eventually. You do, and it becomes quite easy to spot.
You have to sort of train your eyes, one of those things.
Very close to your face, and you look like a bit of a maniac sitting in this grass or staring at this flower, and his dog walkers going by like, what are you doing?
Just so engrossed in the flower head.
Exactly.
Oh, brilliant. Oh, amazing. Thank you so much. So while you guys have all got your questions coming in, I've just got a couple of links for you regarding the talk and the series in general. So I'm just going to pop them over in the chat for you. Now, here we go. And I've also got a link to our questionnaire. If you guys would love to fill that out, that would be brilliant, as it helps us improve and get feedback on these events so we can try improve them in the future. So the questionnaire is just coming through on the chat for you there. Okay, excellent. So I know that we had a couple of questions come through while you were doing your talk. I hope your throat's okay, by the way.
Yeah, sorry about my coughing.
Absolutely
I'm
fine.
recovering from, I completely lost my voice a few days ago, so I'm just really glad that people can hear me again.
Honestly, you've done brilliant in that case, so absolutely no worries.
Sorry, everyone.
Cool, so I think we've got our first question in the chat already. Neil asked, how does the height of the grass affect the quantity of caterpillars found breeding? Or breeding?
Animal breeding. Yeah, this is a really good question. So I don't have any results yet to share, but I can tell you what my intuition says, because I've just finished suction sampling for the year. And it does seem to be that the really, really short grass, there's nothing there. But once it gets quite tall, I would say over 20 centimeters, I wasn't really finding much, to be honest. And I'm not sure why this might be. It could be that it is swamping out other interesting species of plants that these caterpillars also like to feed on, because it tended to be the more diverse patches of plant flora that I was surveying. I was finding more and more species, even though they only really feed on grass. So I can't really give you any solid answers, but I do think there is something to it, that we will be able to find some patterns of what sort of preferences they have for grass. So we'll see. I'll keep you posted. Within the next couple of years, I should have some results coming out that I can share.
Excellent. Thank you, Esme. So Roger asks, how do you take the temperature of the butterfly?
Oh, yeah, sure. So with the caterpillars, I have this, it's essentially a long wire, and that's connected to a big sort of reader thing, big electronic thing. And you very, very gently press the wire. It's called a probe or a thermocouple to the sort of surface of the caterpillar. And you need to be very careful because obviously they can be quite soft and you can accidentally pierce them, and you don't want to do that. And that essentially tells you on the little box what temperature they are. So yeah, you just need to be quite careful because it's such a sensitive probe. You can't touch the caterpillar with your hands because you're really nice and warm, and that might warm them up, and then you get a really warm reading and you're not sure what's going on. So you have to sort of follow them around with it as they're trying to run away from you, trying to take their temperature. I get the impression that maybe the very long, thin needle-like probe reminds them of a parasitoid wasp, because often they will turn around and they'll try and fight the probe back. You feel their sort of little mandibles chewing against it. They're quite brave little critters, really, but often they just try and wriggle away, and they're surprisingly fast. So it's a little bit of a challenge, but often, you know, if I've got a little helper with me, they're sort of helping to guide where I am, and we just try and take the temperature as best we can.
Wow, so what I'm learning is you have not only an incredibly good eye, but an incredibly steady hand as well. These methodology is really quite impressive, Esme. I'm not sure I could be doing this, but yeah, I'm very impressed. So, and I believe we've got a couple more come through on the chat. Okay, so next question. Is it known whether a warm February with a potential earlier egg hatching date will lead to a higher percentage of small blue larvae committing to pupation in July and trying to fit a second brood in that same year rather than overwintering as a fully grown caterpillar, or does the percentage of summer pupators stay about the same regardless of temperature?
That's a really good question. And actually, that is one thing that we did not collect data on, was differences between broods in the year, because you can get a sort of partial second brood in the sort of warm south with small blues. That isn't something that we really focused on. So it's an interesting idea. And I definitely think that needs to be an area of future research, is what's going on between these different generations within a year. So I'd definitely be interested in looking into that. I couldn't really tell you, to be honest, at the top of my head. There's a possibility that it could very much be affecting what's going on if they're emerging earlier and earlier. But chances are, because they're so limited by their food plants, if they're emerging too early so that they then have time for the second brood, they may emerge before the flower is ready for them to lay their egg, for the caterpillar to hatch and to start living inside the flower. I think that's more of the risk, really, would be this sort of breaking down of the relationship between the plant, the flower being ready with its seeds, because that's what they feed on, is the developing seeds inside the flower, and the actual butterflies coming out themselves.
Right, exactly. It's all to do with the phylogeny of the timing between the two is like so crucial. So Neil asks, does the density of the nettles affect the caterpillar, and do they prefer the center or the edge of the nettle patch?
That's a very good question. And again, I haven't had time to dig too much into the data, so I can't give you a definitive answer. But this is something we're looking at, very much so the size of the patches as well, because every now and again you find like a single nettle out on its own, and there's never anything on it, except a few times I have found red admiral caterpillars on these nettles that are all completely on their own, often on the edge as well, red admirals, it seems. And they're a migrant species, so they're, you know, just sort of starting to breed in the UK. Perhaps this is because they prefer a slightly warmer climate. They prefer to be on the sunnier side of nettles, whereas peacocks and small tortoiseshells, they live sort of at the top of the plants. They're already kind of in the sun, but my impression is I don't see them along the edges anywhere near as much as I see red admirals along the edges. But I don't have any formal data to back that claim up with. That's just my feeling from, you know, collecting this data over the last couple of years. Again, in the next year or so, I'll be able to share actual proper papers and things with everyone about this.
Amazing. So we'll have to find more about that, because obviously the red admiral are migrant over from the Mediterranean weather, so it's sort of looking for maybe more temperature that's sort of similar to that sort of sense.
Exactly.
So then we've got our next question is from Peter. Any idea if rabbits feeding on the vetch prefer or avoid flower heads containing the butterfly larvae?
That is a wonderful question. And actually, I have a bit of a strange answer to that. Is what I've discovered is that the rabbits don't actually eat the flower heads per se. They just nibble them off, and then the flower heads fall down and they rot on the ground. And the poor caterpillars are so reluctant to leave their food plants that I have a feeling they probably just lie there and die, unfortunately, as sad as that is to say. It's frustrating that the rabbits will destroy the flower and not even eat it. That is frustrating. Whereas the sheep will just munch the whole thing. They love it. And they don't care if there's anything in there. So I don't think rabbits are avoiding anything to do with the caterpillars. And to be honest, even if they did notice these caterpillars inside there, I don't think they've got any chemicals that taste bad in them. They don't produce any toxins that I'm aware of. I think they'd probably snack on them as well, because it's a nice little bit of protein for them. So unfortunately, no, the rabbits are not on our sides for that.
Oh no. Okay, brilliant. I think that might have been our last question. Let us know if you have any more.
Q&A bit. Should I
Yes.
type an answer to them, or would you like me to answer them now?
Last of all, in the chat, sorry, Neil says, look forward to seeing the results of your research. I thought I'd just read that one out.
Thank you very much.
You're absolutely right.
We'll be doing some outreach through the museums. You can hear all about it if you come on in.
You're absolutely right. There are some more questions in the Q&A box. So coming from Anonymous, I have garlic mustard in my garden on which orange tips lay eggs. Do you have any data on the characteristics, position, etc. of this plant that would attract more egg laying?
Fantastic question. And I have data on this, but I haven't had a chance to look at that data yet. I've been focusing more on species like the small blue and the nettle feeding ones for now, but there probably will be, because from what I've noticed, that the orange tips are laying on a lot of garlic mustard, but they're not laying on all of it. So there must be a reason why they are picking certain plants. So I will be looking into this. This is definitely a topic I'm looking for. And if you wanted to ever look, you might already know, so in which case you can just ignore me, but anyone else, if you wanted to look to see if there are any orange tips laying on your garlic mustard, you need to look at the seed pods at the top, those long, weird, thin things. They're these tiny little sort of almost vase-shaped orange eggs that you can look for, and they're really obvious. And then the caterpillars are super easy to spot because they just sit out there completely in the open at the mercy of the environment. So they're very easy to spot if you're ever interested in looking to see if there's anything laying on your garlic mustard.
Perfect. All these more clues on how I can spot all these butterfly eggs. I'll have to like get my magnifying glass and start searching all these plants. That'll be brilliant. So our next question from Adam. Do you believe that there has been any impact on the results from early on in the study, from the late 2000s to the recent 2020 results, in terms of such as breeding density and adult abundance that can be related to climate change?
Well, we didn't see many, well, actually, this is quite interesting. We didn't really see any particular trends with temperature. So that tells me that it is unlikely to be a consistent change with temperature, like a gradual increasing in temperature. It doesn't seem to be gradually changing the adult abundance or breeding density. But if you look really closely at the data, at some of, for example, that really up and down peak and valley plot I was showing you, one of those peaks and valleys falls over a year where Europe had a massive heat wave in, I think it was 2003, 2004. There was a really bad heat wave in Europe. I'm not sure if any of you guys remember it. And what that did is it caused a massive decline in kidney vetch flowers all throughout Europe. So we saw patterns the following year of lots and lots of these small blues laying loads of eggs on only the few remaining flowers all throughout Europe. It wasn't just in the UK. So these patterns are being seen everywhere, and they may also be being driven by the plant density and the plant's abundance, which unfortunately we don't have data on, or at least not yet that I've looked at. I've got a little bit of data that we'll be looking at in the future, hopefully. But kidney vetch is a very sensitive plant. It really likes dry soils, but it's not a very good competitor. So it's often swamped out by everything else. It doesn't last very long on sites, which may explain why they keep appearing and disappearing, this species, because without kidney vetch, the small blue can't exist in the UK.
Perfect. Thank you for answering that, Esme. Our last question is from Alison, and I'm so glad someone else is asking about this cannibalism, because I have to say I am very intrigued. Alison asks, is cannibalism a necessary survival strategy in some years?
This is a really good question again, and something that I don't really think anyone has ever looked at before, probably because it's very hard to record. You have to sort of sit and watch, I guess. It would be very hard to spot. But I don't personally think it is a necessary survival strategy. It's possible that potentially in some years, if their flower that they're on does not have enough seeds, they could supplement their diet, I guess, with their neighbor or something like that. But they tend to never leave the one flower they're on. If you remember that picture I showed you earlier, where it had two small blue caterpillars, those flowers were right next to each other. They were both large. Neither one of them had left their flowers right next to each other to go and snack on each other. So that sort of makes me think that it's more that, oh, this is my flower. There's not enough seeds for two caterpillars to survive. So I'm going to eat you. I'm the bigger one. So I will survive, sort of thing. So I guess in some senses, yes, it is a necessary survival strategy just to stop competition for these yummy little seeds that are developing inside these flowers that aren't that many seeds to go around. But specifically for it to be cannibalism, I'm not really sure. That'd be a really difficult one to investigate, but I'd make a fun few weeks trying to work that one out.
There you go, Alison. Are you up to the challenge? That is the question. The research is unproven, so you're welcome to go out and search for it then. Okay, excellent. Let me know if you have any more questions. We'll just give it a couple more moments just in case anything else is coming through. So just going back to the chat again, I think we've got another question come through. Oh, can you give us some sample publications? So any of the publications that you've been working on with your research or relevant to the small blue in any way?
Yes, I can. So my paper on the small blue is open access, so anyone can find that one. You just need to search my surname and it should come up. But if not, shall I quickly grab a link and put it in the chat, or is there an email or something we can share? Evie, it's totally up to you.
I've just put a link in the chat now.
Oh, thank you very much, Ed.
Perfect, Ed. Thank you for having that.
Yeah, that's open access. If there is any other particular paper that you would be interested in, please do reach out to me, because, you know, I believe everyone should be able to read whatever publications they want, really. And I'll see if I can help you out and send you some more details, send you some links or some PDFs. So just let me know.
Esme, are you happy with me sharing your email on the chat?
Yeah, of course. Yes. Perfect.
Okay, I'll drop Esme's email and you can get in contact with her directly if you're interested in finding out more on her research. Okay. There you are. Perfect.
Great, thank you.
Thank you so much, Esme. I've really enjoyed that talk, especially hearing about all your different methodology. I think that's been a really fascinating part. Thank you so much for joining us again. Lots of comments and saying really excellent talk. So we really do appreciate that.
Thank you very much, everyone. I hope you all enjoyed it. And yeah, with my email address, feel free to shoot me any questions you might have in the future. If you ever feel like a nice day out, you can also email me, and maybe if I'm around in the field, I often take volunteers out as well with me, so I can show you how to look for some of these caterpillars yourselves.
Brilliant. Thank you so much again, Esme. So that's it for this week's Researcher's Story. Next week will be our final talk in the series. It will be from Dr. Jeremy Thomas. He will be discussing his research into the factors that led to the extinction of the large blue butterfly and how this knowledge has then helped to lead to its successful reintroduction 50 years ago. So that will be on next Wednesday on the 8th of June at 7 p.m. same time. So thank you all so much for coming. I think that's it for us for today. Hope to see you all again next week. And thank you one more time to Esme for the wonderful talk today.
Thanks for coming along, everyone.
Thank you.